Well, friends, today we will discuss the nature of electron transport chain. In earlier video, we have seen there is a generation or reduction of NAD as well as FAD, which is produced during TCA cycle as well as during glycolysis, and that NAD as well as reduced FAD is to be utilized for the generation of ATP. For that, we'll have to understand what is the nature of electron transport chain, which acts as the carriers for the electrons, and we'll have to discuss first of all what is this chain, what it is composed of. As you know, we have seen that this respiration, aerobic respiration, is taking place inside the mitochondria. TCA cycle is taking place within the matrix of the mitochondria. Mitochondria has a membrane, outer membrane and the inner membrane. Within the inner membrane, especially in the foldings, which is the Christie area, there are present electron transport carriers, which is a series of electron protein carriers, or we call them as redox compounds, arranged in a specific sequence as per redox potential. So those complexes are arranged in order of increasing redox potential from negative to positive. And as we have mentioned that these complexes are located within the inner mitochondrial membrane of crista. And these carriers, electron transport chain, the carriers are universal in all organisms. If you will see the carriers, there will be broadly 5 types of membrane-bound electron carriers, which include flavoproteins, cytochromes, copper atoms, ubiquinone, and non-heme iron sulfur proteins. We will discuss them one by one to understand their function and their nature. This chain is also called as the cytochrome system because it's prominent, there are present cytochromes. In addition to this chain is also named as a respiratory chain because 70% of the oxygen taken by the cell is utilized by this chain during oxidative phosphorylation for the production of ATP. So we have seen that there are a sequence of carriers in ETC. These sequence of carriers was worked out by Bertrand Chance and his co-workers. They used number of the inhibitors like amytol, rotenone, and pyridine, which blocks the flow of the electrons. And while using these inhibitors, they understood the nature of the electron transport chain. And also by disrupting the mitochondrial inner membrane with detergents, these complexes were identified. There are 4 complexes of ETC arranged in 4 membrane-bound protein complexes. So there are 4 separate complexes which are identified by Roman numerals from 1 to 4. Let us discuss them one by one. We have a complex 1 to 4 which participates in the electron transport system. So that means these are the electron transport carriers and they participate in electron transport. Therefore, these complexes 1 to 4, they lack stable contacts. They are away from each other. There is no permanent contact between them, but electrons are transported between them by diffusion of mobile carriers. We have a mobile carrier there, coenzyme Q, cytochrome C, which acts as electron shuttles. And you see these complexes, which are 4 complexes there, they contain numerous polypeptides, subunits, and they have prosthetic groups also like FMN, iron sulfur, cytochromes, flavin mononucleotide, iron sulfur, and cytochromes. So one more complex have been identified, which is named as the complex 5. Basically, complex 5 participates in the final process, that's the oxidative phosphorylation. We will discuss these complexes one by one. Before that, let us understand this diagram. We have a mitochondria here, which is having an outer membrane Then there is present an inner membrane which is folded in the cristae, and this inner membrane, if you see, has these electron transport carriers, especially in the cristae area. This is the matrix area, this is the cristae area. So we have the matrix side, we can say the C side. We can say the matrix side, or we can say the intermembrane space side, because this is the space of the between the two membranes. So we have this membrane, which is the inner mitochondrial membrane. This is the matrix side, and this is the inner intermembrane space, or the C side. So we have these complexes which we will discuss one by one. The first complex is this, which is called as NADH dehydrogenase complex, or complex first. It's having within it is having number of redox centers which are arranged in such a fashion that the electrons will flow between them, and the energy will be released. That energy will be harnessed for the pumping of protons from matrix side to the C side. So there are present FMN, iron, sulfur. We will discuss them one by one. There is also one mobile carrier, which is coenzyme Q, which is responsible for shuttling the electrons between the systems. And then likewise there is present the second system, which is called as FADH complex, which is also called as succinate dehydrogenase. And then there is the third complex, which is this cytochrome BC complex, and is called a cytochrome BC1 complex, or complex third. There is a flow of electrons. There is also the pumping of the protons towards this side. There is one more carrier, which is a mobile carrier, that is cytochrome C. And then there is a third, fourth, this complex, that is the complex this, the last complex, which is having the copper as well as cytochrome A, cytochrome A3 terminal complex. It is responsible for carrying these electrons ultimately to the molecular oxygen, and with the help of the protons forming the metabolic water. Also, it is responsible for the translocation of protons towards the C side. And then you will discuss the fifth system, which is fifth complex, which is called as the ATP synthase complex, which has a tunnel through it. There it is responsible for the influx of the protons from this side to this side, from C side to matrix side, which will be responsible for the generation of ATP towards the matrix side. We will discuss. So we have seen in a nutshell here that this area, cristae area, is having an electron transport system which is comprised of 4 complexes, and the fifth one is this elementary particle, which is responsible, which is also called as ATP synthase, that is responsible for the synthesis of the ATP. And there is a generation, there is this matrix side and the C side. There will be translocation of protons from this side to this side to generate a proton motive force. We will discuss it in the subsequent slides. So we have the first complex, the complex 1. This complex 1 is also called as the NADH dehydrogenase, or it is called as NADH ubiquinone oxidoreductase complex. So it is a polypeptide. It's composed of more than 40 polypeptide chains located within inner mitochondrial membrane. That's the first membrane, first complex, which we have seen in the diagram. One side faces towards the matrix and the other embedded within the membrane. It's a major flavoprotein tightly bound to one prosthetic group, namely FMN. Flavoprotein can be flavoprotein internal, that is in FF internal, or it can be an flavoprotein external, which is mostly present in case of the plants. So we have seen here that complex 1 is basically a polypeptide. It's a major flavoprotein, and it has a prosthetic groups which is in the form of FMN, as well as it has a iron sulfur protein. We will discuss them one by one. First of all, FMN, flavin mononucleotide, is a prosthetic group which is derived from vitamin B2, also called as the riboflavin. FMN has fused 3 ring structures. If you see the structure of it, it is this 3 ring structures which has a side chain here. The side chain is ribitol phosphate. So this is the ribitol phosphate, and FMN accepts and donates 2 protons and 2 electrons. So whenever there is an FMN, flavin mononucleotide, with NADH plus H positive, it will give us the reduced form of an FMN and FMNH2 and also NAD. So that means it has a structure, chemical structure, such a chemical structure that it will accept 2 protons as well as 2 electrons. So FMN and iron sulfur acts as redox centers in the nitrogenase, the first complex, NADH dehydrogenase complex, for carrying both electrons and protons. So this is the nature of the FMN, how it is reduced to FMNH2. Then we have iron sulfur proteins. These iron sulfur proteins are basically non-heme proteins. That is, they don't have the pyrrole. Tetrapyrrole is not present there, which contains both iron and inorganic sulfur atoms. There are 4 sulfur atoms on cysteine residues of the protein. They are classified according to the number of iron and sulfurs. There can be dimeric cluster, there can be tetrameric clusters, and some have only iron and sulfur. So this is the structure of the iron sulfur, which is tetrameric. This is dimeric. So we have a protein. This is protein, and there is present in the sulfur containing amino acid cysteine. So sulfur is having with this iron. So there is a bonding between sulfur and this at several points. Here you have 4, which is tetrameric. Here we have only 2. But whole, how many irons will be present there, but this unit acts as 1. So iron sulfur cluster can accept or donate 1 electron at a time, thus classified as 1 electron redox system. There may be number of irons, but it will accept only as 1 electron and donate 1 electron. And it is iron atoms changing back and forth between this state and this state. So ferric and ferrous state. This is about the iron sulfur protein. So these are the prostatic groups which have a role to play in the transfer of the electrons as per the potential from one to another carrier. Summary of this complex is that this complex 1 is responsible for oxidizing NADH H positive to NAD. So we call it as an NADH dehydrogenase complex also. It is responsible for passing 2 electrons to FMN, iron sulfur redox centers, and finally to ubiquinone molecule, which is a mobile, or coenzyme Q, which is mobile within the phospholipid layer. So it can receive NADH plus H positive. That can be from different sources. We have seen one source, that is from TCA cycle, and there can be beta oxidation, there can be malate aspartate shuttle. So these are the sources for NADH, but it is passing on the electrons from this to ubiquinone, and also at the same time it's releasing an energy during electron transfer. That energy is harnessed by this complex to translocate 4 protons. So during this process, 4 protons are pumped to intermembrane space. This is about the complex first. Now let us see the ubiquinone, which is also called as a coenzyme Q. This ubiquinone or coenzyme Q is a small mobile lipid soluble electron and proton carrier. It contains a long hydrophobic chain composed of 5 carbon isoprenoid units. So because of that, it's present within the phospholipids. It has a mobile nature. It's located within the inner mitochondrial membrane. In relation to cytochromes, quinone is present in large quantity. So we have 5:1 ratio because it acts as a quinone pool or a junction point in ETC, receiving electrons from several sources and passing them to cytochrome sequence. So quinone is proportionately more in ETC because it has a role as a postman. it has to carry different electrons from different sources and giving it to the cytochrome sequence. UK ubiquinone accepts and donates 2 electrons and 2 protons. So let us see, first of all, ubiquinone is reduced by 2 electrons and 2 protons to ubiquinol. So we have a coenzyme Q or ubiquinol, which is reduced by accepting 2 electrons and 2 protons. Then this ubiquinone, which is reduced now, passes on 2 electrons to complex third. By passing second complex is not involved here, and translocate 2 protons to inner mitochondrial space. So let us see more details about this ubiquinone. So oxidized quinone form of coenzyme Q, that is this, which is ubiquinone, can accept a single electron, that is when once it accepts a single electron, form a semiquinone, that is this, it has accepted 1 electron to become a semiquinone, and then a second electron, and 2 protons to form fully reduced dihydroquinone. So this is dihydroquinone. This ubiquinol or dihydroquinone diffuses through phospholipid bilayer to donate electrons to complex third. So ubiquinone has a mobile nature and it passes on 2 electrons to iron sulfur complex of third, iron sulfur of complex third, and pumps 2 protons to intermembrane space. So coenzyme Q or ubiquinone is regenerated back, which plays a role in Q cycle, because it is to be recycled so that there is next movement of the electrons, and ubiquinone is soluble in phospholipid bilayer. Because of this, it has a mobile nature and is capable of lateral diffusion. So let us understand now the complex third. The complex third, which is also called as cytochrome C oxidoreductase or cytochrome BC1 complex. What this complex contains? It contains iron sulfur, non-heme iron sulfur, 2 types of cytochromes, the B565 and B560 in plants. There is also a membrane-bound cytochrome C1 and C. C is a mobile carrier. Cytochrome C is a small peripheral protein. It transfers a single electron from complex third to complex 4. In plants, cytochrome C547 is the functional equivalent of cytochrome C. So we have cytochrome complex, which is the complex third. These cytochromes are basically heme containing proteins. So they have a tetrapyrrole chelate of iron, which can exist in ferro and ferric oxidation states. So this is the structure. This is 1 pyrrole, 2nd pyrrole, 3rd pyrrole, and 4th pyrrole. Iron is at the center. So there is a pyrrole. It is called as a heme type of a protein. So they can accept or donate a single electron at a time. Ferro is oxidized form of heme, and this 3 ferric is a reduced form. So it can accept an electron, it can donate an electron. So this is the structure of a cytochrome, which is in a position to accept 1 electron or 1 proton at a time. And it has a structural and functional similarity between, we have seen already electron transport chain during photosynthesis in chloroplasts, where there is a similarity between cytochrome BC complex and ubiquinone with plastoquinone and cytochrome CBF complex of photosynthetic electron transport chain. So what this complex did? Complex third translocate 4 protons per pair of electron. 2 protons from ubiquinone pool and 2 from matrix to the intermembrane space. So 2 from this reduced that's ubiquinol and 2 from the matrix towards the intermembrane space. Now there is a fourth complex which is also called the cytochrome C oxidase, is a terminal complex. This complex contains copper ions in two states, A and B, and two groups of, so there are two copper ions, copper A, copper B, and two groups of cytochromes, namely cytochrome A and A3. This complex receives electrons from mobile cytochrome C, as we have mentioned already, transfers them to heme of the cytochrome A. We have bimetallic copper center, copper A. The electrons are then transferred to copper B through heme of cytochrome A3 and finally to oxygen. We'll discuss it in the diagram. In addition to pumping of two protons, in addition to this is pumping of two protons, this terminal oxidase complex bring about 4 electron reduction of molecular oxygen on matrix side to form 2 molecules of metabolic water. We will discuss it in the next slide. So 1 electron is transferred at a time from mobile cytochrome C through dimeric copper center. A situation is here. The first electron will be taken by the cytochrome A, heme of the cytochrome A. It will be passed on to copper A, and from there these electrons are passed to heme of the cytochrome A3 because it's in vicinity, and to a adjacent copper B, which is 5 angstrom apart. So the electrons are ultimately, pair of electron will be taken by this. One more electron is carried and accepted by cytochrome A3, CuB, copper B, and by binuclear center, which facilitates the binding of oxygen. Acceptance of first 2 electrons reduce this first complex, cytochrome A3 and copper B, binuclear center. The reaction is this. So there will be first the heme of the iron of this cytochrome A and the copper, which is together, it will accept 2 electrons. There will be production of this reduced ferrous, this iron state, and then copper will be changed to this position. Once binuclear center accepts a second electron, a molecule of oxygen binds to it. And now this double bond of the oxygen is broken, forming reactive peroxyanion. Subsequently, 2 more electrons are accepted by cytochrome A, CuB, and 4 protons from the matrix. So it will accept 2 electrons as well as the 4 protons from the matrix. 2 molecules of water are formed there. So for each proton removed from matrix, a negative charge in the form of hydroxyl is left behind, contributing to electrochemical gradient across the inner mitochondrial membrane. Protons used for formation of metabolic water are called as substrate protons. Protons translocated across the membrane are called as pumped protons. This complex provides oxygen, electrons to oxygen, which is an ultimate electron acceptor, protons for formation of metabolic water. It also pumps 2 protons from matrix side to the intermembrane space. So the complex fourth has an important role. It is responsible for the formation, production of metabolic water, as well as the translocation of protons from matrix side to the C side. Let us discuss it. We have a situation here. This is the complex first. This is the complex second. This is the complex fourth, complex first, complex third, and complex fourth. So here, this is a NADH dehydrogenase complex where electrons will be passed on to this, 2 electrons. It will be carried by FMN, then iron sulfur, and these electrons will be taken by the coenzyme Q At the same time, that coenzyme Q will accept the protons and will get reduced to ubiquinone. So this system, once there is a movement of electrons, the energy released there is also harnessed in the pumping of protons from matrix side to the intermembrane space. 4 protons are pumped from matrix side to the C side. This is the complex first. Now we have a Q cycle here. Electrons will be passed on to the cytochromes of the iron sulfur of complex third. But the coenzyme Q is again produced back, regenerated back. That will carry the cycle as a Q cycle, and it also releases its 2 protons, which is from the quinone cycle towards the intermembrane space. While as this complex third also translocates 2 more protons. Electrons are passed on to the iron sulfur, heme, then from this to the cytochrome C1, and one by one electrons will be carried by the cytochrome C, which can only carry 1 electron. As I have mentioned, they can carry only 1 electron, and that electron is, since it is a mobile carrier, it will give it to the complex fourth, where there are present copper in 2 forms, copper A, copper B, and cytochrome A and cytochrome 3. They are arranged in such a way that there is a movement of electrons, as I have already mentioned, that there will be movement of electrons one by one and reduction of copper to its reduced form, and then also there will be the production of water, because those electrons will be accepted by the molecular oxygen, which is an ultimate electron acceptor. In presence of the protons, there will be formation of metabolic water. But at the same time, this complex also translocates or pumps protons from matrix side to the C side. So this is when first complex, third complex, and fourth complex is involved. There is one NADH. It will pump 10 protons, 4 here, 4 here, and 2 here. This is when the 3 complexes, that is the first, third, and fourth, is in coordination. We have a second situation where there is the complex second, that is also called the succinate dehydrogenase, or it is called as succinate coenzyme Q reductase complex second. Succinate dehydrogenase is the only enzyme in the TCA cycle which is embedded in the inner mitochondrial membrane. So it's present within the membrane, inner membrane of the mitochondria, and its substrate oxidation site is located on the matrix site. It contains a group of non-heme iron sulfur proteins which facilitate the passage of electrons from succinate via FADH2 into ubiquinone pool. And it's also a major flavoprotein, as we have seen in case of an NADH dehydrogenase, which is a major flavoprotein. This is also a major flavoprotein, but this is embedded. It's only one enzyme which is located within the membrane of the inner membrane of the mitochondria. And it also has a major flavoprotein. It's a major flavoprotein of the mitochondria, tightly bound to a prosthetic group FAD, flavin adenosine dinucleotide. So we have an FAD. What is FAD? FAD has a ribitol phosphate D-ribose adenine. It has a 3-ring structure. There is a side chain. That side chain has the ribitol. It has a phosphate, and there is D-ribose, and then there is an adenine. So it receives electrons and protons from succinic acid and passes them to ubiquinone. Succinate plus ubiquinone will form a fumarate. We have seen it in case of TCA cycle, but there is a reduction of ubiquinone to ubiquinol. Basically, this only translocates the electrons, no protons are translocated. So FAD will be reduced to this FADH2. This is its reduced form. So we have seen the complex complex second, which is also called as a succinate dehydrogenase complex. Now, what is the role of this complex? This is the complex second, which is also named the succinate dehydrogenase complex. This is, as we have seen, the complex third. This is complex 4. We have seen here that the succinate forms the fumarate, it gives protons, and then FADH is produced there, that is fed to this, to FAD, flavin, FAD, and which is be passed on, electrons will be passed on to iron sulfur protein, and from there it will be passed to the quinone pole. So no translocation takes place here. No pumping of protons when the FADH is fed to the electron transport chain. But here you will see the rest as we have seen in the system third and fourth of the first diagram. Same way, two protons here, and this will pass on single electrons. Molecule of metabolic water will be produced, oxygen will be used there. This is when one FADH is fed, there will be only pumping of 6 protons. These are the 4, these will be 2. So that means succinate dehydrogenase also is responsible for pumping of the protons, but there will be less pumping, that is 6 protons. We have one more complex, which is called as the complex 5th, which is also called as the ATP synthase. In this video, we will only see the structure of it, and in the next video, we'll discuss the function of this complex. ATP synthase is a mushroom-shaped protein complex. It is comprised of 2 major components, namely spherical at F1 with 90 angstrom dia and a basal part as F0 particle. Head is a peripheral membrane protein complex projecting into matrix, as we have seen in the first diagram, is just like an elementary particle in the mitochondria, and F0 is embedded within crystal membrane of the mitochondria, not in the overall inner membrane, only located in the crystal side of the inner membrane of the mitochondria. Both these subunits are connected by central and peripheral stalk. F1 head, being the catalytic portion of the enzyme, is composed of 5 different polypeptides. These are 5 different polypeptides, alpha, beta, gamma, theta, and all these are the proteins of this mushroom-shaped protein complex. 5 polypeptides are highly conserved in many organisms. There are 3 alpha and 3 beta subunits, which are arranged alternatively within the F1 head, resembling the segments of an orange. There are 3 catalytic sites for ATP synthesis, one on each beta subunit. We will discuss this in the during oxidative phosphorylation. So we have seen that this is a membrane, this is the matrix side, and this is the C side. This is the matrix side, this is the intermembrane space side. So towards the matrix side, this is the F1 particle, and which is embedded within the membrane is called as the F0 particle. It is made up of 3 alpha, 1, 2, 3, 3 beta, and then there are also proteins, and the central hub is there, which is connecting it through this. And then there is also present at the base number of these proteins, which are ABC, which are arranged in such a fashion, and these proteins, and then there is also one stalk which is from the membrane, which is having B1, B2 stalk and joined with this protein. So this is the overall structure of a whole complex, which is called as ATP synthase, which is located within the plasma membrane of mitochondria. We will see how it works, what is the function of this complex, and more details of it will be dealt during oxidative phosphorylation. So we have seen we have a complex which has an F1 comprised of these proteins, F2 comprised of these proteins located within the membrane inner membrane of the cristae. So this has a central hub which is asymmetric in shape. It runs from outer tip of the F1 head through the central stalk and connects with the F0 base piece. F0 particle is an integral membrane protein, as I have shown, comprised of 3 different polypeptides, namely A, B, and C subunits. These 3 subunits together form a protein channel or a tunnel. The subunit's composition is A1, B2 because there is B1, B2 as a stalk, and C can be from 9 to 12 segments. The proton channel lies at the interface of the C subunits. Another connective which exists between F0 and F1 through B1 and B2 handle, which is forming a rigid stator that will help to stop it. Rigid stator prevents the rotation of the hexamer. Hexamer is the F1 particle, while it rests on this subunit, central subunit. ETC of animal mitochondria has been studied more intensively than that of the plant mitochondria. So we here in this slide, we have only mentioned the preliminary structure of the ATP synthase. Yeah, it has given an idea that it's having an F1 particle, F1 head, and F0, which is integrated, which is an integral protein within the membrane. And then there is a mechanism having a central rod, and then there is just like an orange, and there are certain proteins in the form of a stalk joined with this head, which acts as a rigid stator. So it's a tiny motor. We will see it in a function. It works as a tiny motor where it is responsible for the oxidative phosphorylation. So we have seen there are 4 complexes, basically 5th complex which has a role in oxidative phosphorylation, because there is the influx of the protons, and those protons will be responsible for oxidative phosphorylation. For that, this complex is involved. What are the evidences in favor of the ETC? We have seen their complexes are basically they are located within the phospholipids of the membrane, and membrane phospholipids are essential for ETC functioning. So if you remove phospholipids, ETC activity will not be there, especially at cytochrome oxidase level. So that means they are located within the membrane. If you remove the phospholipid, ETC function will be not there. There will be no movement of the electrons. Addition of these lipids, it will restore such activity. That means ETC is working. You can make it functional as well as non-functional. In addition to this, there is a flow of electrons in animal ETC which can be interrupted by inhibitors, several inhibitors. We have inhibitors as amytol, rotenone, and pyridine. These are the 3 inhibitors which inhibit different complexes, but we can check that there is no movement of electrons. It has been seen experimentally also. Amytol is a barbiturate which blocks the NADH dehydrogenase. Rotenone is a root extraction of tropical plant. This is the tropical plant from the roots of which is taken, and it tightly binds to NADH dehydrogenase, blocks electron flow to ubiquinone. So rotenone is an inhibitor of NADH dehydrogenase complex, basically taken from the plant species. Its root is a root extract, and rotenone is basically poorly absorbed, especially by the mammals, and it's used for fish catching by South American Indians. But now it's used as an insecticide also. Being poorly absorbed, it is relatively non-toxic to mammals. But now it is used as an insecticide also. Pyridine is an antibiotic and is the structural analog of ubiquinone. So it competes for electrons there. Is an inhibitor of NADH dehydrogenase. These are the direct evidences in favor of the functioning of an electron transport chain. That electron transport chain, which is located within the inner membrane, mitochondrial membrane, especially on the Christie side. In addition to this, plant mitochondria have an additional electron transfer complex, two NADH dehydrogenases, both loosely associated with outer surface of inner membrane. They can oxidize the cytosolic NADH and NADPH and release electrons into ubiquinone pole. So in plants, we have the additional complexes because we have to carry the cytosolic NAD as well as NADPH that is managed by these extra complexes. In plant mitochondria, an alternative pathway exists for oxidizing matrix NADH when complex 1 is either overloaded or inhibited by rotenone or pyridine. So plant mitochondria have basically a rotenone-resistant dehydrogenase for carrying oxidation of NADH derived from TCA cycle. So if you see a plant where from this rotenone is taken, there could be a blockade of its dehydrogenase, but it is resistant to rotenone. So it has been tested in case of the animal ETCs only. What is the output of this ETC now? See, during passage of electrons from NADH, reduced NAD to oxygen, 10 protons are pumped into intermembrane space. A high electric potential gradient develops across the inner mitochondrial membrane. Because when protons are being translocated from the matrix side to the intermembrane space side, the negative charge is being maintained. Hydroxyl will be more in the matrix side. So there will be a potential electrochemical potential difference between the two sides, matrix side as well as the C side. So it is also called this electrochemical potential gradient is also called as a proton motive force, PMF, which of course will be used during the influx downward of the protons from intermembrane space towards the matrix side for generation of ATP or for coupling of ADP with inorganic phosphate to form ATP. So we have seen when one reduced NAD enters the ETC, it will enter at complex first. Total of 10 protons are pumped out. When one FADH, to total of 6 protons are pumped. That I have mentioned in the diagram also. So during oxidative phosphorylation, which will be the part of the next video, we will see that one ATP molecule is generated from the influx of 4 protons. When 4 protons will be pumped back to matrix side through a proton channel, there is a generation of 1 ATP. So that means 1 NADH pumps 10 protons. So there will be 2.5 ATPs generated when 1 NADH is fed to the electron transport chain. Similarly, 1 FADH pumps 6 protons. By that logic, it will generate 1.5 ATP during oxidative phosphorylation. So in the nutshell, during this video, I have tried to convince you that electron transport chain has a function in the movement of in the transport of electrons as well as translocation of the protons. and we have seen the nature of these complexes, functioning of these complexes. In next video, we will be focusing on the oxidative phosphorylation. Till then, goodbye, take care, thank you.